Hi everyone, Dr. Rashid here. In this video, I will be explaining you protein folding and protein targeting. So, coming to protein folding. So, protein folding is a process by which a protein assumes its functional three-dimensional conformation. So, after the formation of a polypeptide chain, proteins are converted into their final three-dimensional configuration. And this three-dimensional configuration is essential for the functional activity of a protein. And this process, that is the protein foldings, are depends on the amino acid sequences and the cellular environment. Cellular environment means the temperature and the pH of the medium. And this three-dimensional configuration is essential for the normal functioning of proteins. So, regarding structural organization, initially they produces primary structure, that is the number and sequences of amino acid. So, this primary structure, secondary structure, tertiary, quaternary, and finally the protein form a three-dimensional conformation. And this three-dimensional conformation is essential for the normal functioning of a protein molecule. And these protein folding processes are regulated by a regulatory protein, and these regulatory proteins are known as chaperones. So, chaperones are cellular protein that aid the protein folding process. So, their functions are they help in protein folding process and they prevent protein aggregation formation. And the chaperones proteins are also known as heat shock protein or stress induced proteins. If there is any defect in this protein folding process, that result in the formation of misfolded protein. So, this protein misfolding can be due to the absence of normal supporting media or deficiency of cofactors, or it can be due to the absence of chaperones protein, or it can be due to changes in temperature or pH. So, let us see the fate of a misfolded protein. A misfolded protein is abnormal one. This protein does not having any function. So, this misfolded protein act as a non-function or a malfunction protein, and their accumulation result in the development of certain diseases. So, failure in protein folding process produces an abnormal protein, that is misfolded protein, which are usually toxic in nature. Let us see some common disorders associated with this misfolded protein, that is Prion disease, Alzheimer's disease and Parkinson's disease. All these are associated with protein misfolding. Coming to Prion disease. So, Prion disease is a protein misfolding disease. And Prion disease, it is a neurodegenerative disease affecting human and animals. So, it is a rare progressive neurodegenerative disease that affect both human and animals. So, normally the neuronal cell contain a protein which is known as prion protein, or it is also known as a normal prion protein. So, misfolding of this normal prion protein result in the formation of abnormal prion protein. So, abnormal prion proteins are toxic and they are accumulated within the neuronal cell and produces degenerative changes within the neuronal cell. That is why it is known as progressive neurodegenerative disorders. So, normal prion proteins are present in neuronal cell. So, when an abnormal prion protein enters into a healthy organism, it induces some changes within the normal prion protein, so that these normal prion proteins are converted into abnormal one. That is why it is considered as an infectious protein. So, prion protein or abnormal prion proteins are considered as an infectious protein molecule, because when they interact with a normal prion protein, they convert this normal prion protein into an abnormal one. And this normal prion protein having alpha helix configuration, and this normal prion proteins are water soluble in nature, but this abnormal prion proteins having beta pleated sheet configuration and they are water insoluble in nature. That is why they are aggregated within the neuronal cell, that will result in neurodegenerative changes. And when this abnormal prion protein interact with another prion protein, that will convert this normal prion protein into another abnormal one. So, they initiate a chain of reaction. That is why they are known as infectious particle. So, normal prion protein, it can be represented as PRPC. Here C stands for common or cellular. So, normal prion protein is PRPC, and this normal prion protein having alpha helix configuration, and the abnormal prion protein can be represented by PRPSC, and this abnormal prion protein having beta pleated configuration. So let's see the functions of a normal prion protein in neuronal cell. And this normal prion protein having antioxidant activity, and they are resistant to oxidative stress. So this normal prion protein prevent neuronal dysfunction. So abnormality of this prion protein produces neurodegenerative changes. So let's see the difference between a normal prion protein and abnormal prion protein. Normal prion protein is PRPC and the abnormal one is PRPSC. So normal prion protein is water soluble in nature. That's why they does not form a protein aggregate. And abnormal prion proteins are water insoluble. And normal prion protein having predominantly alpha helix configuration, whereas abnormal one having beta helix configuration or beta pleated sheet configuration. And this normal prion protein exists as a single subunit, whereas the abnormal one present as a multiple subunit. And the normal prion protein is non-infectious in nature, whereas the abnormal one is infectious in nature. So this prion protein produces, this abnormal prion protein produces neurodegenerative diseases. Let's see the examples for some neurodegenerative diseases produced by this abnormal prion protein. The first one is Kuru, the second one is Creutzfeldt-Jakob disease, and the third one is familial fatal insomnia. So these are the examples for prion diseases seen in human. Come to the second topic, that is protein targeting. So proteins are produced by or synthesized by ribosome. So after their synthesis, the protein undergo configurational changes and they are converted into final three-dimensional conformation, and they are transported into their correct organelle where they produces their function. And that process of transport of protein from ribosome to their correct destination point is considered as protein targeting. So there are two types of protein. One is protein for external secretion, that is cell produces some secretory protein. For example, secretory protein, plasma membrane protein, lysosomal enzymes protein, membrane proteins of the endoplasmic reticulum. All these proteins are considered as the secretory protein, and all these proteins are synthesized by the membrane bound polyribosome, that is a rough endoplasmic reticulum. That is a complex of endoplasmic reticulum and ribosome. Whereas the second type of protein, that is protein for internal parts of the cell, that is mitochondrial protein, peroxisomal protein or cytoplasmic protein, and all these proteins are synthesized by free ribosome molecule. So protein targeting process for this external secretory protein. So these external secretory proteins are secreted or synthesized by rough endoplasmic reticulum, and this polypeptide chain having a specific amino acid sequence at their N-terminal end, and this specific amino acid sequence at N-terminal end is known as signal peptide sequence, and this signal peptide sequence mediate or target this newly synthesized polypeptide into endoplasmic reticular lumen. So this signal peptide present in the N-terminal end of the nascent polypeptide chain target this polypeptide chain into the lumen of the endoplasmic reticulum. So once this newly synthesized polypeptide reaches the lumen of the endoplasmic reticulum, these signal peptides are removed by another enzyme molecule. That enzymes are known as signal peptidase enzyme. And within the endoplasmic reticulum, these proteins are sorted out and labeled, and later some specific amino acid sequences are added to the carboxy terminal end, and this carboxy terminal end amino acid sequence act as a signal molecule for its correct destination. And there are some additional modification like glycosylation reaction that takes place in Golgi body. So these are the protein targeting process for the external secretory protein. Coming to the protein targeting process for internal cellular protein. So the internal cellular proteins are secreted by free ribosome. So this nascent polypeptide chain does not have the signal peptide sequence at their N-terminal end. So this nascent polypeptide chain does not enter into the lumen of the endoplasmic reticulum. They are directly secreted into the mitochondria, cytoplasm or peroxisome. Coming to the disorders due to defective protein targeting process. There are different disorders. Let's see one by one. So the first one is Zellweger syndrome. So Zellweger syndrome is due to defective oxidation of very long chain fatty acid within the peroxisome. So medium chain fatty acid undergo oxidation within the mitochondria, whereas very long chain fatty acid undergo oxidation inside the peroxisome, and it is mediated by some peroxisomal enzyme. In Zellweger syndrome, this peroxisomal enzyme does not reach the peroxisome due to some protein targeting defect. So as a result, there will be defective oxidation of this very long chain fatty acid inside the peroxisome. So they are accumulated within the peroxisome, and later this will lead to the involvement of neuronal cell, and it will produce some neurodegenerative changes, and that condition is known as Zellweger syndrome. And the second condition is known as primary hyperoxaluria. It is due to defect in glycine metabolism, and there is a defect in glycine metabolism due to defective protein targeting for the enzyme, that is alanine glyoxylate aminotransferase enzyme. That enzyme is responsible for the metabolism of glycine, and that enzyme is present inside the peroxisome. So in primary hyperoxaluria, this enzyme is present inside the mitochondria instead of peroxisome. So it is a type of protein targeting defect. And another type of protein targeting disorder is eye cell disease or inclusion cell disease. And eye cell disease is a type of lysosomal storage disease, and it is resulting from defective degradation of glycosaminoglycan inside the lysosome. So these degradations are mediated by certain lysosomal enzyme, and in eye cell disease there will be protein targeting defect for this lysosomal enzyme. So lysosomal enzyme does not reach the lysosome. As a result, glycosaminoglycans are accumulated inside the lysosome, and all lysosome containing organs are affected. So it will produce as multi-organ failure, and that condition is known as eye cell disease or inclusion cell disease. So that's all regarding protein folding and protein targeting. Okay, thank you.